All right, so the next introduction we have is to the International Union for the Conservation of Nature. As you may or may not know, the IUCN is one of the world's oldest and largest conservation organizations. And it's a pretty unique institution because it's a union of both government departments, but NGOs and conservation organizations as well. And all of these organizations work together on various conservation issues. And like all organizations, the IUCN has a vision, which is to have a just world that values nature, as well as a mission statement to influence, encourage, and assist societies through the world to conserve the integrity and diversity of nature and to ensure that any use of natural resources is equitable and ecologically sustainable. So the main way that the IUCN contributes to conservation action and decision making is by gathering and presenting some scientific information in these kind of easy to use packages or tools known as the IUCN knowledge products. So the main IUCN knowledge products are the IUCN Red List of Threatened Species, which you will be learning a lot about this week. They also have Protected Planet, which includes the World Database of Protected Areas. They also have Key Biodiversity Areas, or KBAs, which is kind of just the IUCN standard for identification of areas of global significance for biodiversity. And then they also have the IUCN Red List of Ecosystems, which functions much like the Red List of Threatened Species, but at the ecosystem level rather than the species level. And then the IUCN is also currently working to combine information from all of these knowledge products to develop an index of human dependency on nature, which basically just aims to assess, document, and communicate the role and importance of natural wild resources for a community's food as well as nutrition security. So this training workshop will focus on the IUCN's flagship knowledge product, the IUCN Red List of Threatened Species. And this, like I mentioned, is the oldest knowledge product. It's been around for about 57 years. So for this workshop, we've got three main objectives. The first is to learn what the IUCN Red List is and what a Red List assessment tells us. Second is to understand the IUCN Red List categories and criteria and the assessment process. And then third, learn how to apply the IUCN Red List categories and criteria for global Red Lists. So now that we've got that all under our belt, we'd like to take a few minutes to hear from you. So if you could, I'll kind of go through the list and call on everybody, but if you wouldn't mind telling us your name and your institution, as well as your area of expertise, and then maybe just a few words on kind of your expectations from this workshop, maybe what you plan to do with this training. If there's a specific Red List project that you've got in mind, we'd really love to hear about that. So I'm just going to go across my screen here, and I'm really sorry if I mispronounce your name, but I'm going to start with Pierce Hu. And feel free to turn your cameras on.
Hi, everyone.
Nice
to meet you online. My name is Pierce. I'm based in Yunnan University in Kunming, China. I'm working on butterfly conservation, taxonomy, and diversity distribution across the Himalaya and Henduan Mountains in Western China. My area of expertise used to be taxonomy, but I gradually shifted that to conservation because I realized that butterfly can be a useful indicator for environment health. And I hope to introduce more information of butterflies or moths into the planning of conservation area in the future in my country. That's what I'm doing now. And I hope to learn a few techniques of assessing red list species in this workshop. And thank you.
Thanks, Pierce. Welcome.
Mhm.
All right, next we are going to hear from James Dory.
Hi, yeah, I'm James Dory. So I'm currently at Yale, and I'm a bee person. I'm also at a friend's house, so hopefully it's not too noisy. So I don't really have any expectations for the workshop, but I have been involved in an assessment before, but more as a participant, I think, in some Australian bees after the bushfires. So I'm keen to learn a bit more. Yeah, and also hear about what all of you guys want to do.
Great, thank you. All right, next on the list, we've got Choi Kim Fong.
Hello, everyone. I'm Choi Kim from Forest Research Institute, Malaysia. I'm working on butterflies taxonomy and conservation. And I actually attended the similar course 1 year before, but I missed many sessions, so I hope to catch up those that I missed during last course. Yeah, thank you.
Perfect, glad you can join us. Okay, next on my list is Amin Redzuan.
Hello everyone. My name is Muhammad Al Amin, but you can call me Amin. I'm also from Forest Research Institute Malaysia, co-worker of Dr. Fun Choi Kim, and I don't really have an area of expertise, but I've been working on a few projects on Odonata and also butterflies, and I expect to learn how to assess the conservation status of some butterflies, because I'm currently working on a project that's aiming to set up a few key biodiversity areas in a state in Malaysia. That's it from me. Thank you.
Nice. All right, next we've got Philip Lowe.
Oh, hi.
Hello.
Hi, everyone. I'm Philip, and I'm from Kaduri Farm and Botanic Garden, which is a environmental NGO based in Hong Kong. And what we are doing, conducting a conservation project in South China and the area. And we also conduct quite a lot of rapid biodiversity survey in the area. And my interest is butterfly, and I hope by joining this course I can better contribute in the red list assessment in the future. Yeah, thank you everyone.
Perfect, thank you. Next I'll hear from Michael Orr.
Hey everyone, so my name is Michael Orr. I'm at the Institute of Zoology, Chinese Academy of Sciences in Beijing. My area of expertise, best described as bees, I guess. I do a lot of different things with bees, but recently I've been getting more into biodiversity and conservation sorts of studies, especially looking at things like data biases, etc., because there are so many for insects that we have to deal with. For this workshop, I recently actually put in a grant about a month ago to fix up our database here of all the bees. And part of that, if it goes through, would actually be doing assessments for those species where it's possible. I'm not sure how many it actually would be possible if we really need good data over time, but I may as well at least figure out how to do it so I can better assess whether or not it's possible. So.
Perfect. Thank you. All right, next on my list, I've got Delvin.
Hi, I'm Delvin. I'm from the Mandai Wildlife Group. I'm currently working in the animal care department for invertebrates. So we manage about 82 species of inverts, from their insects to arachnids to crabs, like robber crabs and all. So my expectations from this workshop, so we are conducting quite a number of national level assessments. So I would like to contribute to that, and we're also looking at how we can then use this knowledge to contribute to the global level.
Great. Sounds good. All right, next on the list is Dibjad Yodi. Sorry, I'm mispronouncing your name, I'm sure. I saw you on mute, but we can't hear you. Yeah, sorry, we can't hear you. I don't know if you want to try maybe logging out and logging back on.
Hello, am I audible now?
Oh, yep, now we can hear you.
Okay, good morning, greetings from India. My name is Dibyajyoti Ghosh, and I am from Zoological Survey of India, Southern Regional Center, Chennai. I am working on bee diversity and distributions from Eastern Himalayas, India, and and databases of Indian bees are like to get some information and knowledge from this IUCN Red List thing and see if I can do some justification with the conservation issues in Himalayan bees. Thank you.
All right. Great. And then we've got one last person that has characters that I don't understand. So if you haven't introduced yourself yet, please unmute and let us know who you are. All right. Are you sure you don't want to introduce yourself? One last person. There we go. Hi. I think you're muted.
Am I audible now?
Yep, now we can hear you.
Okay. Hello, everybody. Myself Srijana from Kolkata, Bethlehem College. And my expertise is coleoptera taxonomy in Eastern Indian states, particularly Odisha, West Bengal. And my hope is to get some idea, to gather some idea about the conservation issues of certain coleoptera species in this native area. Thank you.
Welcome. Thank you. All right. Very exciting. We've got people from all over the place. This is wonderful. All right. So now I'm going to stop sharing this presentation and switch it over to a different one. Just double check I get the right one. And I apologize, the first few hours today are kind of presentation heavy, so you'll hear my voice a lot, but I promise in the last few sessions, we'll kind of break out into some rooms and do some exercises and get to know each other a little bit more. All right, so this next presentation is basically just a brief introduction to the IUCN Red List. Hopefully it's up on the screen and everyone can see it. So the IUCN Red List is the world's most comprehensive information source on the extinction risk of plant and animal species. So the Red List uses a sort of standardized listing process that ensures objective, transparent, and repeatable assessments of the conservation status of species at the global level, and it's based on the best scientific information available. Sometimes we don't have very much scientific information available, but we can still try and make an assessment. It's basically just a hypothesis. So species are assigned to one of 9 categories based on criteria linked to population trend, population size, and geographic range. And species listed as critically endangered, endangered, or vulnerable are collectively described as threatened. And the information in the IUCN Red List is widely used to inform and influence biodiversity conservation. So the Red List is kind of the gold standard for informing conservation action. And it's really more than just a name, like a list of names and threat codes. Each assessment includes a species account, which contains information on population size and trends, distribution, ecology, and habitat preferences, whether the species is used by people and what it's used for, what the threats are, and then also current and future recommended conservation measures and research actions. So species assessments are generated through, again, the best available knowledge from thousands of the world's leading conservation scientists and species experts. So each assessment is checked really thoroughly through a review process to ensure that it's really high quality and all of the information is accurate. And back in 2013, the IUCN Red List was officially recognized as an online scientific journal. So that means that all assessments that are published on the Red List have downloadable PDFs that have unique DOI numbers. So with this new journal status, the IUCN is now encouraging assessors to write their assessments at the same quality that they would for any other scientific journal. So you'll notice if you look back to some of the assessments from the 1990s, they're not quite up to this standard, but hopefully when you look at some of the more recent ones, more and more we are hitting that standard quality. So what are we trying to do? The goal of the Red List is to provide information and analyses on the status, trends, and threats to species in order to inform and catalyze action for biodiversity conservation. So traditionally, the role of the Red List was to identify particular species at risk of extinction. And while this is still a very important focus, it's kind of expanded a little bit to encompass the use of the Red List data for multiple species analyses in order to identify and monitor trends in the status of biodiversity as a whole, and to hopefully catalyze or sort of optimize appropriate conservation actions. And to achieve this goal, the Red List aims to establish a baseline from which to monitor the change in status of species, provide a global context for the establishment of conservation priorities at the local level, and monitor on a continuing basis the status of a representative selection of species as sort of biodiversity indicators that cover all the major ecosystems of the world. So what does the Red List actually tell us? The assessment itself is basically just an estimate of extinction risk. So it classifies species by their risk of becoming extinct and highlights those species that are most likely to become extinct in the near future, given current knowledge of population trends, range, and habitat availability, as well as population size and structure, and recent, current, or projected threats acting on the species. So one thing to keep in mind, the Red List is not necessarily just a list of species with the highest priority for conservation action. Just because a species may have a high risk of extinction, it might not be the highest priority for receiving conservation action, and that's because there's a lot of other factors that need to be considered, such as financial cost, as well as cultural, social, biological, and ethical considerations that will make a certain project more effective. And it's also very important to remember when doing red list assessments to ensure that these factors that I just mentioned are not influencing the listing. So as an assessor, it's important not to have any preconceived notions of how threatened a species should be or which category it should be listed under. Remember that it's an objective process, so we don't want to compromise that objectivity by introducing such biases. So who is involved? The red list is compiled and managed by the IUCN. And as I mentioned earlier, the IUCN is one of the oldest and largest international conservation networks. It was formed in 1948 and has been creating red data books and red lists in one form or another for more than 60 years. And the union also works on other issues related to conservation of nature in addition to the species work that's done. And it's made up of, this says 1,300, but it's probably more than that by now, member organizations, and that includes states, governmental organizations, and NGOs. There's more than 1,000 staff located all around the world, and at least 16,000 scientists and experts that form the different IUCN commissions. And there's 6 commissions within the IUCN, and the Species Survival Commission is the largest and works directly with the production of the red list. So if we look a little bit more at the Species Survival Commission, it consists of thousands of volunteer members, including experts and specialists located all over the world. And the membership of the SSC is grouped into specialist groups. So each specialist group either has a taxonomic or thematic focus. Most of them are taxonomy focused, but some are thematic. And moving forward, there's kind of a new specialist group forming now, which is a regional or national specialist group. So that's something we have to look forward to. So these experts provide the knowledge and data that go into the red list and work closely with the IUCN red list unit to review red list assessments. So the SSC is headed by a chair. Right now it's John Paul Rodriguez, and its work is guided by a steering committee. And the work of the SSC receives technical support from the Global Species Program, which includes several thematic units, such as the red list unit, which we'll learn a lot more about. And then others focusing on other species conservation related issues, such as invasive species, trade and use, and one for freshwater as well as marine ecosystems. So who is responsible for compiling the information and carrying out these assessments? Well, really anybody can carry out a red list assessment. You don't even really need to be a scientist or a member of the IUCN, but you do need information from the entire global range of the species you're assessing. So in practice, most of the assessments are carried out by members of the IUCN Species Survival Commission Specialist Group Network. So folks like you all. And this network involves more than 19,000 scientists who volunteer their time, data, and knowledge to help the IUCN compile these red list assessments. There's also a red list partnership that involves a range of different institutions who help fund and manage the red list. And that includes the database, the website, and assessment projects around the world. You'll notice our organization, the Albuquerque Biopark, is one of these red list partnership organizations. And then the IUCN Red List unit in Cambridge has sort of overall responsibility for managing, updating, and publishing the IUCN Red List. But it's very much a collective effort of thousands of people all across the globe. So a little bit more about the IUCN Red List unit. The Red List unit has been around since 1999, and they were built specifically to compile and manage the IUCN Red List of Threatened Species. This slide might be a little bit old, but back in 2020, there were about 10 people working at the Red List unit. Not all of them work on Red List assessments, but certainly these 3 people across the top are people that you might come into contact with if you submit any assessments to the Red List. And the first is Craig Hilton Taylor, the second is Caroline Pollock, and the third is Janet Scott. So again, these are the 3 main people that you're likely to come in touch with at the Red List unit. So the Red List unit does a lot of different things, but mainly what they do is manage the database. It's called the Species Information Service, or SIS, and we'll learn a lot more about how to log in and use that service in the coming days. And then they're also responsible for updating the Red List website at least twice each year. They're also responsible for developing and maintaining standards for IUCN Red List assessments. So some of the documents we'll be using over the next few days, the guidelines, the documentation standards, they're responsible for all of those. They also review all of the assessments that are submitted for publication on the Red List. And this involves checking that the categories and criteria have been applied appropriately, proofreading, ensuring assessments have been thorough, and that they've been appropriately peer reviewed. And they're also involved in various other conservation activities associated with the Red List, kind of either directly or indirectly, kind of technical support and advice for a host of different things. These are some other things that the Red List unit does. They develop all of the training resources for Red Listing, including the presentations that Moni and I will be using over the next few days. They also develop tools for compiling and managing Red List data. They manage the IT infrastructure and provide technical advice. They develop indices for monitoring the status of biodiversity over time. In particular, the Red List Index, which is an important tool for reporting on the status of biodiversity. And if you're interested about the Red List Index, Moni is a really good person to talk to because it was developed in collaboration with a few different Red List partners, including BirdLife International and ZSL, where Moni used to work. The Red List unit also provides support for global and regional assessment initiatives. They do work with key biodiversity areas. There have also been incremental in help developing the green status of species, which is a new product that the IUCN launched last year. So they do a lot, and they're only a team of 10 or so they're kept very, very busy. So how can Red List data be used? Red List data are used by a lot of different sectors to inform and influence biodiversity conservation. So it's used in analyses to develop indicators and influence policy and conservation decisions. So for example, the IUCN and its Red List partners regularly publish analyses of the Red List data, informing us of the current status of the world species. And we can show you where some of these analyses live on the Red List website. It's also an important tool used to inform conservation planning and priority setting. So for example, if you want to decide where a key biodiversity area or a new protected area should be put, you can pull up Red List data and find out where the highest concentration of threatened species is in a certain region. So that spatial data is available on the Red List. It's also been adopted by several international conservation policies and agreements as an indicator of the status of biodiversity. So Red List data is used to measure progress towards many of the Convention of Biological Diversity targets. as well as some of the Millennium Development Goals, which now I think have been updated, but let's see, what else? It's also an extremely important tool used for driving funding mechanisms. So there's a bunch of different conservation funds that rely heavily on red list data to determine whether or not the species that people are trying to get money for are threatened. Many corporations and businesses consult the red list when making key business decisions. And then one of the most important roles of the red list is as a communication tool. So it highlights the importance of biodiversity and healthy ecosystems to the public, the private sector, politicians, etc. But it also doesn't just focus on threatened species. It can be really useful to highlight maybe near threatened species, or I run into this a lot with invertebrates, data deficient species. Where do we need research? So it can be a really helpful tool to draw attention to some of those issues. And then this last slide, this is just to bring this tool to your attention. It's called the Integrated Biodiversity Assessment Tool. And it's basically just a way to access some biodiversity data from a few different sources. So it's a collaboration between the IUCN, BirdLife International, and some other organizations. And it was designed to be a sort of one-stop shop search service for those seeking information on global biodiversity. So you can pull information about from the world database on protected areas, as well as the red list, as well as key biodiversity area information all at once, and download it through this tool. So it's kind of a neat tool to be aware of. And that brings me to the end of that presentation. And I think I've got one more before we take a short break.
Are there any questions so far from anybody? While Anna is pulling up the next presentation.
Is everybody asleep already? Let's see.
It seems like everybody's happy. Ooh, yes, probably a question coming up. Yeah, feel free to unmute yourself or or raise your hand, which I think is under the reactions button, or you can also just type your question into the chat if you prefer that. So that's various ways. So far, everybody seems happy, and James is not quite sleepy yet.
All right, so this next presentation should be pretty quick, from raw data to red list. So this will kind of just go over the red list assessment process. So first of all, let's talk about what is a red list assessment. So again, just to reiterate, an IUCN red list assessment is basically just an estimate of extinction risk. So the red list highlights those species that are most likely to become extinct in the near future, given current knowledge of population trends, range, and habitat availability, as well as population size and structure, and recent, current, or projected threats acting on the species. So before outlining the actual process, let's look at the final outcome. So kind of what we're trying to achieve. And this example here is for one of the most data-heavy species that's probably been assessed on the red list, the African elephant. So we certainly don't expect every assessment to be this thorough, especially those of us who will be assessing insects. Unfortunately, we rarely have an assessment with as much data and content as this one. So red list assessments are published on the IUCN Red List website, and they include the red list category and criteria code, and also information about the species. So in this first part of the species account, we can see we've got the scientific name as well as the common name of a species. A lot of insects don't have common names. Don't fret, you don't need to include one. We also have the assessment citation and then a photo of the species. Again, only if we have one, though this can be really helpful for communication purposes. So we do try very hard to get photos, hopefully live photos of most of the species that we assess. Doesn't always happen though. And then we also have this visual illustration of the red list category that the species is listed in. And then over on the right, we've got the assessment date with a little download button that I'll come back to in just a minute. We've also got a distribution map and a summary of the coded habitat types there on the bottom left, as well as the threats and conservation and research actions for the species. So then there's this button over here that says skip to assessment in detail. And if you click that, you will be taken to a more detailed section of the assessment. So it's kind of further down the page. And in this detailed section, you'll see the taxonomy of the species. Oops. And then you'll also have this justification, the names of the people involved in the assessment. And the justification is basically just explaining what data they've used to show that this species is vulnerable under A2A, for example. And you'll learn all about what A2A means probably tomorrow. So again, the people involved in the assessment are in the lower left here, and then the date and year that the assessment was published. You'll also, if you scan down further, you'll see the whole geographic range, including all of the countries where the species occurs. Then you'll also get information on population and habitats and ecology. And then further down, you'll see all of the threats impacting the species and conservation actions. And then you'll have a full list of references cited throughout the entire assessment. So returning to the top of the online assessment page, this little download button, if you click that, you'll have a link to the PDF version of the assessment. And then also, if there's any supplementary material, which often there's not, but sometimes there is, you can link to that here as well as some other things like if it's been written up as an amazing species, which doesn't happen that often. And then also importantly, you can download the shape files or the GIS range data for all of the species on the red list here as well. So this is what the assessment PDF will look like. And again, the size of the PDF will depend on how much information is available for the species. Some are just a couple pages, and it looks like the African elephant is about 40 pages. And again, the species has supplementary material, but that's not always necessary to include. So a red list assessment has three main components. It's got the red list category and criteria. So for this purple skimmer, it's listed as vulnerable. Vulnerable is the category, and then A2A, B2, AB3 is the criteria. And over the next few days, you will be learning what all of these numbers and letters mean, so that when you see this, you can look and automatically know, okay, this species is experiencing population declines and it has a very small range, and it looks like there's some habitat loss. So you'll kind of learn how to decipher this as you become familiar with the criteria. So then second, you've got documentation supporting the red list category and criteria. So that's things like population size and trend, the range, the threats, the conservation measures, etc. And then you'll also have a distribution map of the species. And if you've never used any mapping software before, we will have a little bit of a demonstration later in the week, but there's also some newly developed tools that should be available soon on the REDLIST website that kind of walk you through how to use even free mapping software like QGIS or Google Earth. So what can be assessed? Now that we know what we're trying to achieve, how do we start the assessment? So first we got to figure out what we're going to assess. And the REDLIST categories and criteria can be used to assess any described taxa, including species. So for example, there's an assessment for the tiger at the species level. You can also assess subspecies. So all of the tiger subspecies have also been assessed. For plants, you can assess varieties. You can also assess subpopulations of species, although keep in mind that IUCN has a very specific definition for what a subpopulation is, and we'll learn about that in just a few minutes. So what about microorganisms? We actually don't include assessments for microorganisms on the REDLIST, so we can't assess those. And undescribed taxa, there are some instances where the REDLIST unit will let you assess undescribed taxa, but only if the species is clearly distinct. So ideally, a description should already be underway. Museum or herbarium voucher references must also be supplied with the assessment to indicate where the types are held. Information must be available on the range of the species. So for example, if all you know about the range is that it was collected from a market stall somewhere, that's not enough information to allow you to assess the species. And then there should also be some conservation benefit to listing the species on the REDLIST. So if it's going to be data deficient or least concern, you should probably just wait until that description paper comes out, and then you can assess it from there. But if it's looking like it might be endangered, vulnerable, that's a good reason to get it on the REDLIST sooner rather than later. So the categories and criteria were developed for use at the global level. So that's all data from across the species' entire global range that is considered during the REDLIST process. But the criteria can be used for regional or national assessments as well. It's important to note that they weren't originally designed for this purpose. So to ensure that they're applied more or less consistently, there is a guidelines for application of IUCN REDLIST criteria at regional levels. I will mention now, we weren't planning to go into these regional level guidelines, but if there are enough people interested, we can kind of shuffle the schedule around and fit it in, or we can do maybe an hour-long session at some point at a later date if that's something that you are interested in. So please let us know if that would be useful for you. So in all REDLIST assessments, we only consider wild populations within the species' natural range. So captive populations and populations introduced for reasons other than conservation are not included in the assessment. So each assessment will reflect the status of the species in their natural habitat. So for example, there's a map here on the slide. All the purple areas indicate where this species, which in this case is the horse chestnut tree, has been introduced outside of its natural range. The species is actually only native to Bulgaria, Greece, and Macedonia down here. These areas in yellow. So only data from those native areas should be considered for the REDLIST assessment of this species. You can still document that this species is quite widespread across Europe, but when you're applying the categories and criteria, you'll look only at this native range. So a lot goes into publishing a red list assessment. So we'll look over the next few slides at kind of what's involved in the process. So sometimes there might be a project manager. So this might be if the assessments are being done as part of a larger project. The project manager will be the one that coordinates the process. So they'll kind of liaise between the various other people involved. They'll sign off on the assessments before they're submitted to the red list unit, that kind of thing. There aren't always project managers, but with larger projects, there might be. Assessors are usually the species experts who provide data and knowledge to inform the red list assessments. So they're the ones who will apply the red list categories and criteria to assess species based on available data. And again, anyone with sufficient knowledge of a taxon and a good knowledge of the IUCN red list categories and criteria can do a red list assessment. And assessors come from both within the IUCN network and from outside partners as well. There are sometimes contributors, so these might be other experts who provide data or knowledge, but are not necessarily involved in applying the red list categories and criteria. So they don't actually assess the species, but they do contribute data to the assessment. Let's see, then we've also got reviewers. Reviewers are very important. Each red list assessment must go through a review process before being published. So again, this is kind of just to ensure that all of the data is accurate and up to date. And the reviewers are generally experts from a red list authority, but not always. And basically, as you're writing these assessments, your specialist group will help you determine who's going to be the reviewer on the assessments that you produce. And then the last step, or the last people involved, are the IUCN Red List unit, and they'll manage the Red List database and coordinate the publication of assessments on the website. So once you're finished with your assessment and it's been reviewed by somebody, you'll submit it to the Red List unit and they'll do a final review before it's published on the Red List. So as you can imagine, completing and publishing a red list assessment takes a bit of time. So the process involves first literature searches. So we're going to be looking at all published as well as unpublished scientific literature and reports, internet searches. And if you're not yourself a species expert, you interviewing species experts, or if you are a species expert, maybe just checking with other species experts. Sometimes workshops are held, and this is basically to get all of the different species experts in a room to kind of make sure that they agree on all of the data that's being used. And then all of the assessment information will be entered into a database. Again, it's called SIS. And once the categories and criteria have been applied to the information, the draft assessment is ready. And then the draft assessment will need to be reviewed again. Somebody from your specialist group can point you to a reviewer. And then from there, the assessment goes to the Red List unit for a final quality check. And eventually it is published on the IUCN Red List website. So once assessments are submitted, the Red List unit looks for errors in spelling, grammar, and formatting. So these are typically things that they can just fix without having to get back in touch with the assessors. So pretty easy. They'll also look at whether the account makes sense, and if they can't really understand what is written in the account, they'll contact the assessors to provide clarification. They'll also be looking at does the species account fully support the final assessment? So if some of the supporting information for the final assessment is missing, again, they'll contact the assessors to request that that additional information is added. And then probably most importantly, they'll look at whether the categories and criteria have been applied appropriately. And if there's any indication that they have not been applied appropriately, again, they'll contact the assessor to try and work through some of the concerns. They'll also double check for the full list of references and data sources cited in the account. And then if there are too many issues to fix before the next red list update, the assessment will be returned to the assessors for resubmission later, which would be a bummer. So there are a lot of procedures to go through before an assessment can be published. Luckily, there's quite a bit of guidance to help assessors and project managers through the process. So the first document that you'll want to become really familiar with is one that I mentioned earlier, the IUCN Red List categories and criteria. So this is just the basic rule book for carrying out any red list assessment. So it has definitions of all the terms used and also lays out all of the thresholds. Then we also have the guidelines for application of the IUCN Red List criteria at regional levels, which again will be specific to regional red listing projects. And all of these documents can be found on the Red List website as well as in that Google folder that we connected you with. The next important document is guidelines for using the IUCN Red List categories and criteria. There's also this really nifty one-page summary sheet that I have on my desk at all times, and it's just a really great quick reference of the different criteria as well as the thresholds that need to be reached. And there's documentation standards and consistency checks. So this is more just some of the formatting details, how are citations formatted, a lot of other kinds of information, kind of the nitty-gritty details can be found in this documentation standards document. There's also a rules of procedure, which outlines the Red List assessment process. So kind of some of the things we just discussed, what are the roles and responsibilities of the Red List Authority, what does the review process look like, what does the petitions process look like, that kind of thing. And again, this can all be downloaded from the IUCN Red List website, and a little bit later in the week, we'll go through exactly how to access all of that information. Any questions?
There's a question in the chat from, oh, I lost it now. From Shui Kim. Sometimes newly described species have as limited distribution information available. Should we still, are we still encouraged to assess those species? That's the question.
Yeah, I mean, it depends on the species. If it's from a place that, if it did have a large distribution, you might have come across it sooner. Maybe it would be worth assessing it. Maybe it is vulnerable. But again, it's kind of just a case by case. And honestly, the Red List unit can be a little bit picky about which undescribed species they're willing to accept, because I've certainly run into it before where I had a fairly good case to list one and they didn't let me. So now I'm having to reach out to species experts to make sure that there's a description underway. So it's certainly worth reaching out to the Red List unit and inquiring whether it might be worth listing, but I don't know. Moni, do you have any experience with undescribed species?
Yeah, I mean, this one was newly described species. Sorry, I don't enunciate properly. Yes, yeah. So I mean, there is, I mean, in many ways, what we're trying to encourage people to do is with species descriptions to already kind of start thinking about what is the information that would then be needed to go into a red list assessment to speed up the process. I know that we've been talking about this for a long time to kind of help some of these newly described species onto the red list. As Anna said, quite often you will have some sort of idea that, well, we've only just described the species, so it's potentially really quite range restricted. So then I would definitely go and prioritize that species for assessment. But if it's something that was newly described, but it's probably quite widespread, we can assess it, but it's probably worthwhile waiting a little bit more until we have better data.
Sometimes you might have information on threats from a region, so there's a lot of things that are going to come into play to kind of help you make this decision. And sometimes it seems like we don't have enough information to assess a species, but if we were stuck in that mindset, honestly, for insects, we'd never assess anything. So we really do rely on inference and speculation quite a bit with invertebrate red list assessments, unfortunately. So,
and also red list assessments should never be, I think quite often we think a species has now been assessed and that's it, but red list assessments evolve, right? Our knowledge evolves, and as we get more knowledge coming in, there is nothing stopping us from reassessing that species with all the new knowledge that we gained. So if we maybe were precautionary with a newly described species, and we're like, oh, there's some threats and it might be quite range restricted, so let's maybe it would kind of sort of trigger a threatened assessment, and then if in future we get more data and see it's not, then we can reassess it and say that this essentially has now been reassessed because of better knowledge. But I personally would always err on the side of, you know, if we can somehow assess it, we should probably best do it. And when I say if we can assess it, that's generally down to having the manpower to do it. It's fair. And there's more questions coming in about endemism, which depends on a species' full geographical distribution range and is tricky to determine. So what to do? I suppose, again, that refers to newly described species, and that's from Pierce, right? I mean, the thing is with the red list, we're not really assessing endemism as such.
Well, Moni,
yeah, there you are.
I entered the text just to answer the previous question. What about endemism? Because I think it's very tricky to determine whether it's endemic species or not. Depends on the region or country you discuss.
Yeah, precisely. It's always relevant to what endemic to what area, right? And this what area, that's the thing that defines if it's an endemic or not. And the red list doesn't really assess endemism, it assesses extinction risk of a species. So it could be covering multiple countries or 2 countries or several regions. It could still be a threatened species. So we're looking at extinction risk rather than endemism.
Yeah, right.
Cool. The other thing that I've found is a few thumbs up for regional assessments at regional level in the chat.
Yeah, yes, sure.
Yeah. And then we've got 2 more people who joined us who might want to very quickly introduce themselves. Evrim, I don't dare to ask what time it is where you are.
It's nearly 5 a.m. 5:11. Hello, everyone. I'm sorry, I'm a little bit late.
You want to quickly introduce yourself and where you're based, what you're working on, what you're hoping to get out of this workshop?
Hello everyone. I'm Evrim. I'm from Turkey. I actually one of the writers of the butterflies, Red Data Book of the Butterflies of Turkey. It was a national red list, and it was 10 years ago. I think 12, 13 years ago. I'm one of the species survival commissions of butterflies in Turkey, and we currently want to work on the new red list in Turkey. So I thought that this would be a good place for a refreshment, as many things have changed. So I'm glad to be here, and I'm sorry I was a little late.
And technically you're really early, I think. Just look at it like this. And then we've got Damika as well from Sri Lanka, I believe.
Yes. I think you can hear me now. Okay.
Yes, yeah, we can hear you.
Okay.
Excellent.
Okay. I am Damika from Sri Lanka. Actually, sorry for late joining because I had some problems in the internet connection. Anyway, so I am working with the Firefly Specialist Group. So I'm working on fireflies in Sri Lanka. So, and actually I am glad to participate this program anyway. Thank you very much.
Wonderful. Excellent. I think it's time for a very quick little break. I don't know, Anna, I can't quite see the, where's our...
Yeah, we're a little bit ahead of schedule, but that's fine. I think we will take a 15-minute break. Does that sound good to everybody?
Okay, thank you.
Great. We'll see you in a bit.
Well remembered.
I thought I better do it while I was thinking.
That's very true. I would have forgotten. Right. How are we all doing? All refreshed? I assume everybody is back. It's difficult to tell. See you, Ephraim. Cheers.
I'm here.
Good, excellent. Piers is there. Delvin's there. Excellent. There seems to be some movement. Phil, perfect. I hope you all got a chance to have, like, I don't know, a cup of tea or something refreshing. I shall start first of all by sharing my screen. Can you all see this? Yeah, good, excellent.
Yes.
Perfect. Let me see. Yeah, good, excellent. Fabulous. Shall we kick off again, Anna? Sorry, I can't see you now.
No, that sounds great. Let's do it.
Good, excellent. Awesome, brilliant. Right. Thanks everybody for coming back. It's always good to have people return after the first break. So as Anna said beforehand, this session today is a lot of us talking at you, sadly, but really a lot of these presentations form the very basis of red list assessments. And I would probably say this next session is very much what forms the basis of red list assessments, because in this session now we're really starting to go into what are some of the key terms and concepts that underlie the IUCN red list criteria. Now, as Anna said, the red list assesses extinction risk. So these key terms and concepts, they're essentially things that are related to concepts or parameters of extinction risk, things related to population size, for example, with small population sizes, potentially meaning that a species is at a higher extinction risk, looking at population declines, looking how spatially distributed a species is, and so on. And so this presentation really is very much the, I would nearly call it the heart and soul of the Red List, but maybe I'm being a little bit overly dramatic. Anna is sort of nodding. Cool, excellent. So this is a little bit a dense presentation. If you've got any questions, please put it in the chat, raise your hand. Anna, please monitor in case I miss anything, any hands being raised. And if you've got any questions, just ask there and then so that we can clear up any confusions. And if I do suddenly speed up and talk too fast, please also just tell me to slow down. Yes, good, thank you. Excellent. So as I said, really these key terms relate to things that, well, things that have something to do with extinction risk. And as Anna already said, when we look at extinction risk assessments, like for this amphibian species here, on the red list, we see the category, in this case, critically endangered, and then we see this character string that is this weird looking blurb that might not mean much to you now, but by the end of, well, actually by the end of tomorrow, you will be able to say what this character string means. And this really, this criteria code tells us a lot about why a species is threatened. So for example, in this case, yes, critically endangered, specifically because in this case, it tells us something about the species having undergone at least 80% reduction in population size. So it's declined quite rapidly over the past 10 years or 3 generations. It also tells us something under the B1 criterion, that is a restricted extent of occurrence. So something about its range that puts it at a higher risk of extinction. It also tells us that the population is potentially severely fragmented or occurs in just a single location. So 2 other concepts that the IUCN Red List looks at that relate to extinction risk. And it also tells us something about there being a continuing decline in extent or quality of habitat. So once we've gone through all of the criteria, this criteria string A2, ACE, B1, AB3 will actually kind of condense what we assess the species under into one neat little criteria string. But these terms that are marked here in red, they're really the terms that we need to make sure that we know what they mean. So what is a reduction? What is meant by population size? What is a generation? What is the extent of occurrence, and so on. So the IUCN Red List generally uses these in a very specific way, and this is what this presentation is going to look at. Now, quite often we're familiar with some of them, but it's worthwhile paying attention because in some cases there's tiny little nuances of how it means something very specific to the IUCN Red List. So Anna already showed you what I also call my best friend, this criteria summary sheet here. As with Anna, I've got this on my desk and it's just my quick little reference to anything Red List related. If you have this already up, or if you see it now here on the screen, you can see that a lot of these terms that we've just seen, but also some other terms pop up all over this criteria sheet. And these terms are very central to the criteria, but you will also see that not all of the criteria use all of the terms. So there's, for example, one criterion that looks much more at population size than another, and so on. So not all of the criteria use all of the terms, and the best thing also is that we don't need to, well, we need to understand what each term means. We do not need to have data available for every term to be able to carry out a Red List assessment. So, for example, quite often with invertebrates, we have distribution data that will help us to start mapping our species distribution and also tell us something about the range extent of our species, or then using the range-based criteria that the IUCN uses and so on. And we might have some additional information on population size that we could use under another criterion, but we don't have to have data for all of these terms. This is also why we have 5 different criteria to help us a little bit with our data availability. So terms used in the IUCN Red List. Let's start with a relatively straightforward one. And that is, imagine that you have here a species of tree. It could be any other species as well. It could be a fungus, it could be a bird, it could be a mammal, it could of course be an invertebrate, like a firefly or a bee or a butterfly. You can see here that this species is split into several different groups. You can also see that there's some unoccupied areas between them. The big trees here, they're generally represent mature reproducing individuals, adult trees, so to speak, and the little ones, they denote baby trees, the ones that aren't quite reproducing yet, they're too young to reproduce. You could also see up here, this arrow here between these 2 slightly disjunct bits of this section up here. This indicates that there is actually movement of genetic material between these 2 groups. So seeds, pollen, and so on. If it was a different species altogether, if it's an invert, it could be movement of adults, for example, between these 2. Now, for the purpose of a Red List assessment, when we talk about population, we mean the total number of individuals of the species that we're looking at. So that includes all adults, all juveniles, senescent individuals, and so on, across the entire global range. That would be our population. More often, though, the IUCN Red List criteria talk about population size. So they don't use the term population, they use specifically the term population size. And this is measured as the number of mature individuals only. So in this case, all of those adult trees here make up the population size of this species. Now, what are mature individuals? Mature individuals, as you can see here, there are those individuals that are known, estimated, or inferred to be capable of reproduction. So again, just reiterating, juveniles, senescent, or repressed individuals are excluded from this parameter. So it's just those that are known, estimated, or inferred to reproduce. And now the reason why we focus on mature individuals is to allow the criteria, first of all, to be applied in a consistent manner to taxa that have very different life histories. And really, by focusing on mature individuals, we focus on the functional part of the population, i.e. the part that is likely to actively contribute to the next generation. Now, admittedly, for quite a few species groups or species or taxon groups, it's quite challenging to define what a mature individual is. I would, for example, I wouldn't even know where to start if somebody asked me assess this fungi and tell me what the number of mature individuals are. But the great thing is that many other people have already come across these slight head scratches and puzzling situations. So the Red List guidelines that Anna pointed out earlier on, they provide more guidance on how to estimate the number of mature individuals appropriately for a large number of different scenarios. So it's a really good resource when you have a species that's a little bit unusual. Another concept that's important and gets used a lot in several of the criteria is that of subpopulations. Now, subpopulations are distinct groups within the population between which there is little demographic exchange. Typically, the IUCN would say it's no more than one successful migrant individual or gamete per year. Now, these subpopulations, they can be separated by some geographic barrier, for example, or by, say, a mountain range or ocean or something, or it could be a large area of unsuitable habitat that the species can't cross between. Or it could also be that there's some sort of behavioral mechanism that prevents individuals from different groups to interbreed with each other. So that could, for example, be the case in a migratory species, birds, for example, where groups of individuals migrate kilometers and kilometers to use the same feeding grounds over the summer, but then when they return to their breeding grounds, they're again in their very distinct subpopulations, so they will never actually interbreed. So that would be another example where subpopulations would be applied. Okay, so that's the first bit. That's subpopulations and population size. Now a little bit more of a closer look at mature individuals. Now I already said that this is the number of individuals that are known, estimated, or inferred to be capable of reproduction. But of course, when we estimate this, we really need to take into account what species we're looking at, the species biology, its behavior, population densities, and so on. And so the number of mature individuals may not necessarily be the total number of adults in the population. It could be that where you have, all adult males or all adult females are breeding, and the number of mature individuals in this case would literally just be the sum of your reproductive females and reproductive males. So you've got 100 breeding adult females, 100 breeding adult males. The population size would be around 200. Now, quite often it's not that simple as that. It could be that you have fewer adult females than the number of males, for example. And then how you might want to estimate your number of mature individuals is by looking at your number of reproductive females, essentially double that to represent their male counterparts. So if you have 50 adult females, 150 adult males, your population size might actually be closer to 100 rather than the sum of both groups, which makes it 200. So it's really important to filter in all that you know about the biology of your species and how it reproduces and who reproduces and so on into your mature individual estimate. Now I should have already clicked on this. Yes, absolutely. In things like social bees and ants, for example, you may, for example, have some mechanism in place that suppresses some adult individuals from reproducing. And then you would also need to take this into consideration when calculating your population size, so your number of mature individuals. So in this case, a large colony, only one or a few reproducing females at any time. New queens can be promoted, and then the mature individuals might be counted as follows in this equation here, where you essentially take your number of active queens times some expression of the number of potential queens, that is a realistic estimate, times 2 to essentially make up for the male counterpart, for example. And again, there's a lot of guidance on this in the guidelines on how to apply the categories and criteria. So the guidelines document really is absolutely a treasure trove of any situation in terms of different species biology that people have so far come up against. And then in conversation with the standards and petition committee, really these guidelines get developed to make sure that under these specific circumstances, the red list categories and criteria are applied as is intended by the red list. So it's a lot of work's gone into this, and it's a really good resource. Okay, enough about mature individuals. We'll move on to the next term, which is that of generation length. So generation length is a concept that's used in red listing to essentially scale our time-based measurements. So for example, our rate of population decline to account for the different rates at which different species survive and reproduce. So to account for different life histories, for example, and different speed of reproduction. Now, the general rule would be the generation length is generally greater than the age at first breeding, and is less than the age of the oldest breeding individual. So it's somewhere in between there. Of course, with the exception where you have taxa that only breed once, in which case the generation length is the age at which this breeding occurs. Now, the reason why we need to look at these, species declines over specific time periods is that we really need to make our population reductions that we might see appropriate for the taxa that's assessed, appropriate to the species that's being assessed. If we think about a short-lived species like, say, a butterfly or a dragonfly or a mouse, if we went into the vertebrate realm, then 10 years is a reasonable time period over which to measure a significant population decline. But when we think of a long-lived species, say a redwood tree or an elephant or a giant tortoise, for example, then 10 years really is a very insignificant time period, and population declines have to be measured over a longer time period that's more relevant to that species. And so this is why the IUCN invokes this generation length a lot in the criteria. Right, yes. Anything else to add here? Nope, that's it. Right. Generation lengths, how do we define it? There are several definitions, they're all acceptable, and I also fully realize that quite often when it comes to invertebrates, we don't really need to think in too much detail about generation lengths, because quite a lot of our species are very short-lived, which is in many ways a bonus, to be quite honest, but so that you know about the kind of things that go into calculating generation lengths, here are some examples. Now there's several definitions, they're generally all acceptable. Really what's the most appropriate depends on the life history of your species. Quite often also it just depends on the data that we have to make an estimate of generation length. If, for example, we were going to study something like, say, birds, for example, where we can identify the mother of each new chick in a colony, for example, and record the age of all of these mothers from the youngest parent that we found to the oldest parent that we found, and then we average all of this across, then this average age would be our generation length. So the average age of parents of the current set of newborn individuals in the population is one of the definitions. Of course, if we really did this properly, we would have to do this probably several years to kind of really find what is the general age distribution of this species throughout the years. So that's one of them. Another one is that's quite often used where people develop things like life tables. That's again quite common in invertebrates, in mammals, for example, is where we follow one group of individuals that are born in the same year and record their reproduction over their entire lifetimes. And each year the ages of all of the breeding individuals from that group that we're following is recorded from age at first breeding to age at last breeding, when they produce their last offspring. And again, our generation length would be the average of all of this. That's another way of going about generation length. A third one is if we know that, for example, typical female of a species produces 6 offspring over its lifetime, then at what age 50% of that offspring produced. So at which age will it have produced 3 of them? And that would then be the age, the generation length for that species. So there's various different ways. There's the mean age of parents in a population at the stable age distribution, or the time required for the population to increase by the replacement rate. There's different definitions. They're all very in detail described in the guidelines. Most of them you will probably realize are not that relevant to invertebrates. For invertebrates, quite often all we need to ask ourselves is when we look at the criteria tomorrow, is however many generations we need to look over, is that I don't know, longer than 10 years, for example, to figure out over what time span we need to assess our species. So quite often we don't have to worry about generation length at all. But for some criteria, it is very important to keep this in mind, because this scales our time-based measurements to account for these different survival and reproduction rates of different species. It reflects essentially the turnover rate of breeders. And the other thing we need to always consider is that we always use the natural generation length. So for some species of fish, for example, in response to exploitation, they will start reproducing earlier. Now, this is not what we should generally use. We should use the natural generation length that we see when there's no pressure on this species. And again, there's a lot of special assistance that the IUCN has to estimate generation length based on some survival rate and fecundity for different species. Again, this is very much focused on vertebrate species, these workbooks. I personally have rarely used them, and lots of guidance in the guidelines. Right, now we're getting to the terms that are much more frequently used when we speak about invertebrates, for example. So first of all, we're now looking at stuff that relates to population reductions. So the first one is simply, the term is reduction. Very original indeed. And a reduction is simply, when the IUCN mentions a reduction, it's simply a percentage decline in population size over a specified time period. For example, you will see this in criterion A, for example. And here you can see that this specific time period is 10 years or 3 generations. So this reduction is something that we scale depending on the generation lengths of our species to make it appropriate for our species. There again is a tool available to help us use any data that we have available to calculate a reduction. I think we've put them all into the folder that Anna shared, and it's also downloadable from the REDLIST website. But really, this is quite a good source, because this is quite a good tool, because it lets you put together data from kind of disparate data sources and try and figure out what your decline rate would be based on the data that you've got. So that's really quite useful. And again, also the guidelines, always worthwhile checking the guidelines to see what they have to say about how to calculate reductions, particularly in specific cases. But for now, really, you just need to think reduction, percentage decline in population size over a specified time period. And this is in contrast to another term that looks at essentially a decline, which is continuing decline. So that's another term the IUCN uses a lot. And a continuing decline is a recent, current, or projected future decline, which is going to continue unless we actually do something about it. So unless some remedial measures are being taken so that we could reverse the decline. Now, on first glance, they probably look quite similar, but there are 2 main differences between reduction and continuing decline. Now, the primary difference is that a reduction, for example, can just be a one-off event. or it could be ongoing. So this is different to a continuing decline because very much by definition, a continuing decline is expected to continue. So that has to be ongoing. A reduction can be a one-off event, like you had a volcanic eruption that wiped out part of the species, and then afterwards the species either remains stable or kind of increases again, bounces back. Whereas a continuing decline is something like that's being brought about, for example, by continued levels of exploitation, for example, that we see. So there's nothing, unless we stop exploitation of this species, this decline is going to continue. Secondly, also a reduction, when we talk about reduction, we only ever talk about a reduction in the population size. So in the number of mature individuals, whereas a continuing decline can apply to, yes, population size, but also a lot of other parameters that we're looking at for the purpose of red list assessments. So is there continuing decline in some of the range-based metrics that the IUCN uses, extent of occurrence and area of occupancy? Is there a continuing decline in the area, extent, or quality of habitat, in the number of locations, in the number of subpopulations, for example? So these are the main differences between these two. Okay. Our whistle-stop tour of IUCN terms continues, and we go on to extreme fluctuations. Again, remember that all of these terms relate in some sense to the extinction risk of a species. Extreme fluctuations, of course, increases species risk of extinction if there are these wide, rapid, frequent variations in population size or in the number of subpopulations or locations that we see. And so a species with extreme fluctuations is important to highlight during red listing. So what we mean by extreme fluctuations is that we have these rapid variation, frequent variation, which is typically a 10-fold increase or decrease in either the population size or the number of subpopulations or the number of locations, or in some cases also the distribution. So where you have species in, say, freshwater species that are in flooded habitats, where the habitat can be enormous one moment and then kind of shriveled up to nothingness again the next moment, for example. Here's an example. It's also an example of a butterfly, so yay, which is great. Imagine this species of butterfly where the number of mature individuals in the population changes significantly, as you can see here, from year to year. So if you look from July in year 1 to July in year 2, where it's suddenly less than about 2,500 total individuals after it's been up to 20,000, to the year 3, where it's between 10,000 and 15,000, and then it shoots up to, well, 17,000 again, drops again the year after. Then this is a species that shows extreme fluctuations. As you can see, that's real changes in total population size from year to year, if you go from July to July. This is in contrast to this species. This is an example of a killifish that relies on temporary ponds. So each year during rainy season, when the pond that this species is in, it fills up. It stimulates the species to develop eggs, to have the development of eggs into adult fish. The fish swim around. You see a lot of these mature individuals, they're reproducing. But then at the end of this season, the ponds dry out again and the dry season approaches. Now the adults will then die off, but they will have also reproduced and left eggs behind. And so you could say, well, you know, from year to year, we go like rainy season, lots of them, dry season, none of them, because we don't see the mature individuals. But really here, what we see is that the actual population hasn't really changed in size. it's just a change between different life forms that we've seen. And then the next year again, if we go to the next rainy season here, the species bounces back. And really, if we kind of look at it, this is just a natural seasonal fluctuation. It's not anywhere near tenfold increase or decrease that we would need to see for an extreme fluctuation. And hence we wouldn't call this an extreme fluctuation. So this is not an extreme fluctuation, and the previous example was an extreme fluctuation. I think we're probably all quite happy with that. Okay, you're doing really well. Oh, there's a hand raised, I think, Evrim.
Hi, Moni. One question is that an extreme fluctuation might be very natural for that species, as you were talking about the butterflies, right? So in that situation, do we still consider it as a parameter for red listing? You know, the butterflies always fluctuate, right? I mean, that is naturally a tendency for butterflies to fluctuate like that. So this is not a surprising graph for me. So would that be a really important criteria?
And so extreme fluctuation, yes, it doesn't matter if that's a natural fluctuation. If you think nothing is going to get assessed just because it shows extreme fluctuations, this term is only ever used in combination with other things that increase the extinction risk. And so what you're looking at here is really something that, for example, has already shown a dramatic decline, and is then on top of this also fluctuating. So it could essentially fluctuate itself out of existence, or has a restricted range where there's only probably a relatively small population size. Same thing again, it could easily, particularly in the presence then also of a threat, just fluctuate itself out of existence. I like using that term, but yeah, so it doesn't matter if this is a natural fluctuation. Sure, that's what it does. And this is probably because, I don't know, different climatic conditions from one year to the other, or the host plant is doing something from year to year, for example, that impacts this fluctuation of the butterfly, and that can be absolutely natural.
Okay, thank you.
That makes sense. No problem. Right, where was I? Cool. Excellent. Severe fragmentation. You're all doing really well. I think we're more or less nearly halfway through all the terms that we need to discuss. So stick with it. So severe fragmentation is actually, well, I was going to say it's pretty straightforward, but actually it's one of the terms that probably is often not correctly applied in red list assessments. So severe fragmentation relates to our population of our species. So we see severe fragmentation in a population when most individuals, in this case more than 50%, because that's the majority, is found in small isolated subpopulations between which there is very little dispersal. So these subpopulations may in fact be too small to be viable. Now, the reason why I say that this is quite often not properly applied is that quite often people, assessors will say, well, the habitat is clearly fragmented, so this species has to be fragmented too. That's obviously not really the case. It depends very much on whether dispersal is possible from one of the patches to the other, and whether it's actually also occurring. So here are two examples. It's not just enough to have these small isolated subpopulations. And if we consider, for example, this taxon with a highly mobile adult stage, for example. So adults can happily travel from one patch to the other. And that just means that they can essentially rescue or recolonize sites where the population is declining, or even where it's gone extinct. So in this little animation, because of the dispersal ability, for example, these patches where the species has been declining or even gone extinct can be recolonized. This is very much focused on things like metapopulation theory, for example, this severe fragmentation. Now, this is in contrast to species that that are not very able to disperse, or if we were talking about, I don't know, plants that produce small numbers of pollen or anything that can disperse. And hence this species, because of the lack of dispersal ability, is more easily isolated. So we've got the same setup in this case, but if these small patches that are not in themselves viable go extinct, there's absolutely no way in which that species can recolonize these patches. And so it's essentially leading to a population reduction. So the first animation we saw, that's not severe fragmentation because we can have dispersal between these patches. The second animation we've seen, that's severe fragmentation under the definition of the IUCN. But ultimately, that's pretty much all there is to it. So it's quite straightforward. Now we come to my favorites, not gonna lie. This is because quite often for invertebrates, the first thing we have is some sort of spatial data, right? We know where our species is. So some of these range-based metrics are really the first ones that we can calculate during our red list assessment, because it's very much the base level of kind of biodiversity information that we have. So these two range-based metrics that the IUCN uses is the extent of occurrence and the area of occupancy. So the extent of occurrence, really, very simply, is if you look at this, this schematic here on the right, and all of the areas there in dark green or olive green are the areas where the species is known or projected or inferred to occur. And the extent of occurrence really is the area that's encompassed by the shortest continuous boundary that we can plot around these points. A minimum convex polygon really around these points. Now note that there is a lot of area included here that doesn't have the species present, and that is fine because the extent of occurrence is not meant to be a representation of the species range. The extent of occurrence, in fact, is meant to measure the degree to which extinction risk or risk from threat factors has to spread spatially across an area to impact the entire population of a species. So it's very much dependent on how, on spatial spread of extinction risk. So you could think that something that has a smaller extent of occurrence is likely to have a higher extinction risk because threats would spread over that area more easily, you would think. They're closer together. Threats are probably also quite similar. Whereas if you have a large area in between, even if the species is not there, a threat that occurs here on the left might be unlikely to also occur on the right of that polygon. So this is really how you have to think about extent of occurrence. The second one is area of occupancy, and this really is the area within the extent of occurrence which is actually occupied by the species. Now the IUCN measures this by overlaying a 2x2 kilometer grid and then counting the number of occupied cells. That's essentially the way that this metric is being calculated. There's really not much else to say about it apart from showing you an example of what this looks like in practice. So this is a distribution map here of I think it's a dragonfly species. You can see that it's mapped to some sort of hydrobasin map. You can see the red dots, which is where the species has been found. And you can see those blue polygons, which are essentially mapped as the limits to the distribution or the hydrobasins where the species is most likely to occur in. Now, when we look at the extent of occurrence, that really is the entire area around all of these basins where the species is inferred to occur. So this is quite a large area here. I think this is New South Wales and up to Brisbane. So this is the east coast of Australia. And this is a very large range indeed. And again, you can see that there's lots of areas here where we don't have any occurrence records for the species. When we look at area of occupancy, again, this range is far too big to overlay a 2 by 2 kilometer grid system and still see something. But if we zoom into Brisbane up here, really we've got, if you look at point localities that we've got, we would have something like an area of occupancy of 40 kilometers squared. If you assume that the species occurs in all of these hydro basins, in all of those areas, then you would possibly get up to about 400 kilometers squared area of occupancy. Which quickly leads us into something that the IUCN is very keen on. If we are uncertain about our estimates for any of these parameters, it's absolutely encouraged, in fact, to provide a range of these. So in this case, we could, for example, say it's between 40 and 400 kilometers squared based on our two different calculations that we carried out for area of occupancy. So this is a very quick illustration just to kind of, again, hit home how extent of occurrence really measures the spatial spread of extinction risk across an area. So this is actually two setups, comparison of two taxa. They have the same area of occupancy, but different extent of occurrence. And if we look at, oh, there it is, good, excellent. It took a while for the animation to take place. If we calculated the area of occupancy here, we would have 10 squares in which the species is distributed. So same area of occupancy for the one on the left and the one on the right. But quite a difference in extent of occurrence because one of them is much more spread out and the other one is more clumped on the left. Now this really illustrates the effect of having a larger extent of occurrence, but the same area of occupancy. And if, for example, we assumed that these species were impacted by the same threatening event, and this would be a threatening event, for example, that would occur over the area of 6 grid squares, for example, here, and wipe out the species in these squares, then you can see that both species lose area, but of course, this is much more pronounced in the species on the left, where a much higher percentage of area is being lost. Ultimately, if you now look at the area of occupancy, again, there's not huge amounts of change between these two species, but there's definitely, you can see how the extent of occurrence in the species on the left-hand side, has lost a much higher percentage. It's about 1/3. I think that's been lost. Whereas the other one continues to have a large area. So this is again showing that it's very much about how risk or threats spread over an area. This is what you need to consider for extent of occurrence. Okay, some slightly special provisions under extent of occurrence is what to do with migratory species. They must have an enormous extent of occurrence because they've got different wintering grounds and different summering grounds. Well, actually, when it comes to migratory species, we're really only considering the smaller area of either summering or wintering grounds as the extent of occurrence. Say the smaller areas or the smaller minimum convex polygon around these areas. And that is because the species is either in one area or in the other at a time. So it's independently kind of dependent on both of these areas. There's also some things to consider when we talk about area of occupancy. The first one is any of you who work on species that occur in linear habitats, fresh water, for example, something that occurs along a river. There's sometimes a concern that these 2 by 2 kilometer grids do not have much ecological meaning for the species in that linear habitat. for example, and that's fine. It's not meant to have ecological meaning as such. It's meant to be a measurement that's consistent with the threshold values that are given in the red list criteria. So even if a species occurs along a river, we use a 2 by 2 kilometer grid for the area of occupancy estimates. And then the other special provision under area of occupancy is that you have some cases where you get a area that is really essential at a particular stage in the life of a species. That could be, for example, a specific nesting site where an entire colony of a species essentially nests. That's their home base, right? And so this smallest area that would be essential at any stage to the survival of this existing population of a species, that can be measured as the area of occupancy. So for example, if you have a species that's confined to a rock in the middle of the ocean, as this is their breeding rock, and then from there they spread out and feed and do whatever they need to do, and then that breeding colony would be, for example, the area of occupancy. Makes sense. It's the area that's the smallest area essential at any stage to the survival of the species. Okay, we're all still awake. I can only see Ephraim and she's still awake, so I use her as an indicator here. Okay, we come to one of the last, but also one of the funnest concepts in the IUCN Red List, and that's the concept of a location. Now, this is one to pay really close attention to, because this is probably the most misunderstood and misapplied term in the Red List criteria. So the term, when the IUCN uses it, the term location means a geographically or ecologically distinct area in which a single threat event can rapidly affect all the individuals of the taxon. Now, a location is, as you can see, there has to be a threat event. So a location is by definition defined by a threat event that is happening to a species. So if there is no threat impacting a species, we cannot define the number of locations. That's the first thing to remember. There always has to be a threat that we consider, and our number of locations is defined by this threat event and how it impacts a species and over what area it impacts a species. Now, the reason why it's often misapplied is that quite often people think that, assessors think that it means the number of sites or the localities where a species is recorded from. But that's not it. You can have several localities in a single location. You could even have several, or well, more than one, at least several subpopulations in a single location, because location is defined entirely by the threat. Now, the easiest way of looking at this is through a few examples, and I have to say the easiest examples come from freshwater, because everything is so nice and neatly confined. So this is a good starting point to think about location. So we've got this freshwater species that's distributed in a lake. So the black dots are where the species has been found, and it's also in a lake nearby. Now, if we have this species and we assess it, and we know that invasive species poses a threat to this species, then really the number of locations is defined by how many of these invasive species events we need to impact all individuals of our species that we're assessing. And for invasive species, we would need to introduce our invasive into the river, assuming that that invasive is good at swimming upstream as well. If this is like a predatory fish, for example, it will be very capable of doing that. And we would also, that would be our first location, and then we would also have to introduce it into the lake to affect the individuals in the second location. So under the threat of invasive species, in this scenario, we would have two locations, one in the river, one in the lake. Does that make sense? Don't see any hands up. Probably everybody's asleep. Pollution how does pollution impact a river? Well, it generally spreads downstream. If we know that our main pollution source, for example, would be pretty much at the top of this species distribution, and pollution is the most plausible threat that's impacting this species, then we would have everything downstream from this first pollution event. We would have this as a location, and we would also have to have it in the sidearm on the left, the sidearm on the right, and also a separate event in the lake to impact all of the individuals in our species. So that would be 4 locations. Of course, for pollution, it very much depends on where the most likely entry point is, right? Because we could also come up quite easily with an estimate that would come up with 4 to 5 or even more than 5 locations, also a little bit on at what stage do we no longer see pollution effects downstream and so on. So all of these things need to be factored into our assessment of the number of locations. And again, as you can see here, we do have 4 to 5 locations, for example, stated. It could be that we're pretty sure it's between 3 and 5, then we list 3 and 5 as a range. It's absolutely fine in our assessments to introduce uncertainty. Now, what happens if we actually know that part of our distribution is not affected by any threats? Can we still use locations? Well, if most of our distribution is not affected by any threats, then we can't consider locations, because location needs a threat to be present for it to even exist as a concept, right? By definition. What happens if we have a threat in part of the range, but then also we don't know what happens in another smaller part of the range? Well, we can use subpopulations as a surrogate for locations. That might be a good approximation, for example. We can use some estimate of what the smallest area would be affected by the main threat impacting our species to determine the number of locations, and break down a range based on, I know, the size of the smallest area affected, say, by a logging concession, for example, if logging is one of the main threats to our species, and then we kind of parcel up our distribution like this. We can also use a range of different threats. So it could be that a certain threat might not is affecting part of our distribution, but not part of the other. And then we can just use the most likely threat that occurs in the other areas to determine locations there. So in short, we could have a mishmash of different threats to assess the number of our locations. Again, we're using a freshwater system. I'm sorry that it's very freshwater based. It's conceptually the easiest, I think, to visualize areas over which threats spread, because they're so well defined and so kind of in themselves kind of quite small areas. So pollution could be impacting this left arm of the river. It could be also impacting this part of the river. There could be, for example, damming, which affects another part of the distribution of the species, fishing in another. And so we can kind of build up this mosaic of different threats to count our number of locations. In this case, depending on what you do with the various leftover bits of river that don't have a threat assembled, indicated on them, it could be something between 5 and 6 locations, for example. If there is no plausible threat, we cannot consider locations. It's as easy as that. No threat to the species that's impacting the species, we can't define locations, because it always requires a threat by definition. And with this, you've made it through. Oh, there's a raised hand, Evrim. I'm glad at least one is still awake.
Are we allowed to ask complicated questions about location or
Sure.
case studies that we had? Well, we usually have the problem that in butterflies, there's a very restricted range species in one area, one population, and there are multiple threats occurring on it. possibly influencing the same population. A case we had was like there was a small population and there was a construction of road, grazing, and near in the same area there was pollution of pesticides, like the close proximity of pesticide application. So what do we do in a case like that if we cannot really separate the population? Like 20% of this is influenced by that or something like that. It is usually what our minds are confused in a situation like that. Do we need to run a threat analysis or prioritize the threats, or what is your suggestion for that?
My suggestion would be to think about the different scenarios going one threat by the other. It's a range restricted species, right? So you would probably come up with, well, under, say, if it was just pollution and you know that, I don't know, the same pollutant is being, I don't know, pumped out in one part of the distribution but affects all of it, and you might have one location. If you know that there's different pollution events that are needed that impact the entire area, you would probably have something like 5 pollution events needed, so something between 1 and 5 locations or something, and then you would do that for the next threat. That's how I would do it. When I can't prioritize which one is the most important and they're all kind of interlinked, I would probably try and figure out what is the range of locations that I can get from each of these threats, and state the range of locations and say why. If I can't make up my mind which one is the most important one, if there's one that clearly seems to kind of top all of the others, then I would go for that one.
Okay, thank you.
But yeah, quite often, particularly with pollution in terrestrial environments, with impacts of like small scale agriculture that springs up generally around very localized areas, right? It's really difficult to figure out how to parcel up your distribution in terms of threats. So what I generally think about is this a threat that generally occurs over a larger or smaller area? And I would probably then, if they're all equally as likely, I would probably then go in favor of the one that is likely to impact the largest area and use that to define my number of locations. If I can't make up my mind which one's the most important of those, and they're all affecting them, because this then would let me go for a precautionary approach. And ultimately, that is the better approach to take for a species.
Because what usually we come across is this number of locations is very important. And if in one single population there are 5 threats different influencing the species, all of a sudden the number of threats within a population is 5, even though it is only one small population. And then that increases the number of locations, which kind of changes the threat category, but actually it's a small population with lots of threats on that tiny population. And when we divide up the population with the threats, we're actually underestimating the cumulative impacts of all the threats altogether.
So it's really not necessarily about the number of threats, it's about over which area have these threats an impact, right? So how many of these, if, for example, you have a species that's impacted by pollution and by, I don't know, fire, I'm just making stuff up, how many fire events would you need to impact the entire population? When it's in a really restricted area, I would say potentially the way the world works these days, if it's a really, really tiny area, probably just one fire, right? Because it's spreading over the entire area. And so it's really about the area over which a threat impacts a species, again, rather than the number of threats impacting it, if that makes sense.
Okay.
So quite often when something is very, very restricted, then it's highly likely to have, it's not necessarily the case, but it's highly likely to have a small number of locations.
Mhm. Okay, thank you.
Cool, no problem. I can literally talk about locations all day long. Quite to the pain of everybody else who has to listen to me.
Moni, there's a couple more questions in the chat. And so,
Okay.
Srinjana, if you want to ask your question, I know you had your hand up, or I can read it out of the chat. It's up to you. But the first one in the chat, if in the case of sea level increasing, it will affect those populations living in coastal areas. Can we use location in this assessment, or can we apply location in this assessment?
Absolutely, yes. So sea level rise, yes, climate change is starting to really make a, I was going to say splash, that's a really bad pun because it doesn't, in many ways, when it comes to defining the number of locations, because you could, if something occurs in a relatively restricted area and you have a climate change impact, like it's in a low-lying area and you can already see the impacts of sea level rise and maybe increased storm frequency that impacts the species, then yes, based on if that's the serious threat that is going to impact this species, then you could use climate change as your threat that defines the number of locations, and your location might just be one. Which could be the same, for example, with increased fire frequency in a small area, which might be caused by, I don't know, drought, which might be caused by climate change. So you get all of these knock-on effects, but that could also then be used to define your number of location. And depending on what area your species occurs, this could then again, if it's very restricted, it could be one location because you just need one fire and that's impacting the entire area where the species occurs and the species is impacted by it. Or if it's over a larger area, you might have an estimate over what area a usual wildfire, for example, occurs. And you could apply this to figure out how many locations there are for your species. But yes, you can definitely use, well, any threat really that impacts the species can be the one that then defines number of locations. It's just a matter of trying to figure out over which area this threat is impacting the species and how many of these threat events you would have to essentially impact all of the individuals in your population. Do you have anything to add, Anna?
Yes, I will say, so if we're using climate change as our threatening event to define our locations, we just have to be really careful that we specify what the mechanism of that threat is. So sea level rise is a really great one. That's a really clear threat caused by climate change that's going to make it pretty easy for us to define our locations. And I will say too, I have used sea level rise as a threat to define locations. And in this case, the species was pretty habitat specialist, but there were a few occurrences that were a little bit further inland. So I used sea level rise to define at least one of the locations, and then I used a different threat. I think it was maybe urban development or something to define my second location in that specific case. So there's a lot of ways to go about location, and the one thing I can't stress enough is just to be very, very clear how you're defining your location. So what is your major threat? What size of an area does that major threat cover? This is certainly one place where time after time when I'm reviewing assessments, it's just not quite clear how they've determined locations, and I have to go get additional information from them. So it's really good to understand this concept as much as possible.
Yeah, and again, the really most important thing is that each of those concepts, if you use them in the Red List assessment, explain properly why you're using it and how you're using it and what the impact is on the species, particularly when it comes to anything relating to threats, because at least in the past, I think there were quite a few assessments where people were like, oh, it's probably impacted by climate change. Oh, one location. It's like, well, that's not necessarily true. It's true, yes. I like the sea level rise one, because if it's really coastal, then you've got a relatively clear-cut picture. Or if you've got something that's in a small area that is now seeing increases in wildfires because of climate change, for example, that's a very clear-cut picture. In many cases, the picture will be less clear-cut. You sometimes have mountaintop species where you probably can also say that's a pretty clear-cut picture, because there really is nowhere else for them to go. And you've already seen other species migrate up the mountain in response to climate change. Then I would probably also kind of say, well, this species that's already kind of run out of altitude at which to go, then I would probably also be quite happy to go with, sure, that makes sense. But yeah, always justify your assessments as much as possible. It sounds like a lot of work at the time, but ultimately the big bonus is that it goes through review much quicker. And so the more time spent initially is actually, it saves time later on in the process, I'd say. Time well spent.
And there's one more question in the chat. Threats like predation pressure or lack of prey, how could this be considered in the case of location?
So predation pressure, that's a good one. So usually we look at, when we look at threats, we look at threats that are kind of anthropogenic threats that impact a species. Now, predation pressure could, of course, there could be issues with predation pressure because it comes from invasive species. That could be then we could use invasive, for example, if this is what causes it. So we're really, lack of prey, again, what's the underlying issue for the lack of prey? Is more the kind of threat that we're looking at. So it depends a bit on the underlying causes that cause these things. Because predation pressure generally sounds to me like it's just a very natural thing that happens. But why would this suddenly no longer be natural, and why would this suddenly be a threat to the species, I think is the thing that needs to be looked at. I don't know what you have to add to this, Anna.
Yeah, I think that's certainly true with predation pressure. Lack of prey might be a little bit different, because maybe the prey species is suddenly threatened and that puts pressure on the predator species. So you certainly could use that in some way to consider location. I think if that were the case, I would dig into what the prey species was threatened by, and you could kind of use that as your threat for the species that you're assessing. That would be my suggestion.
Yeah, so we had a few assessments where we did assessments of dung beetles, and of course some of them are very dung type specific. When it comes to the African species we looked at, for example, I do remember there were some that were elephant dung specialists, but also confined to some parts of West Africa where, of course, forest elephants aren't doing that great. To be quite honest, in these cases, we very rarely used location at all. We probably inferred something about population declines from the data that we saw in the elephant population, assuming that the dung source is going to dry up, and hence it will have similar knock-on effects to the beetle. Can't think of any other examples where we looked at essentially lack of prey. I mean, this is lack of breeding dung, but yeah, similar. Cool. Are there any other questions? And we will come back to talk about location. I promise you that again and again over the course of this workshop. I think it's fair to say. Just really quickly to finish off, this is the last thing that we very briefly need to at least mention as an honorary mention. Another thing that the IUCN Red List categories and criteria can use to assess extinction risk is quantitative analysis. So that's any form of analysis which estimates the the actual probability of extinction of a taxon based on what we know about its life history, about where it occurs, its habitat requirements, threats, any management that might already be applied to that species or that are options for that species, essentially something like a population viability analysis, but for the entire global population of a species. Suffice to say, I think this has been used very rarely. I don't know what the latest number is, but I know that I asked about a few years, 9, good. 9 species have been assessed under using quantitative analysis. It's a process that's very data heavy. It requires a lot of iterations of going through your models again and again and again. And also it requires a lot of, why is nothing happening here anymore? I froze. No, there we go. It also requires a lot of supplementary material that you submit to the Red List to justify the use of your model, explain how you did the modeling. And this actually then is something that goes not just to the, I would say, the Red List unit, but actually to some people who knows how these techniques properly work to kind of be reviewed by them. And there's guidelines. In the guidelines, there's information again about quantitative analysis. So you can see quite a bit of it. It's been very rarely used. And it's probably something that for inverts, I know in every workshop, somebody's like, oh no, I think we could be doing this. And I was like, I'm looking forward to reading this assessment. It's not happened yet. 9 species, good. 9 species have been assessed using quantitative analysis. That would obviously be like the holy grail, because it gives us a probability of extinction of a species, but we never really, very rarely have the information to run any of these assessments under quantitative analysis. Right, I think we've talked already way too long about terms because I can't stop myself. And we're going into a first little exercise. So Anna will have set up some breakout rooms for you all. Probably working in groups of 2 to 3. I don't know, you set them up, Anna?
Yeah, we're going to do 3 groups. So you should have 3 or 4 people in your breakout room. And if you've never used breakout rooms before, it should be pretty intuitive. Once I click the Open All Rooms button, you are going to click something somewhere that says Join the Breakout Room. And then you'll end up in a room with a bunch of people. Moni and I can kind of shift through. And so when you first get there, don't panic, maybe just reintroduce yourselves and we'll show up at some point to give you the term that your group will be working on. And I still have to assign them real quick. So Moni, you can go ahead and explain the exercise.
Cool, excellent, cool, no problem. So in each of the groups, in each of the breakout rooms, you will be given one of these Red List terms that we've just talked about. And so what we want you to do is within your group, discuss again the meaning of what you just learned about this term. What does it mean? What does it mean in the Red List context? And then in your group, find one example. It could be real, it could be fictional. We really don't mind. That demonstrates what this term means, how you would use it in a Red List context. And really just explaining what it means in your own words and in your own example. And so we give you 15 minutes for this. And then at the end of this, each of the groups will present their example.
Are you ready?
Excellent, good, yeah. I was looking at when you looked like you were about to be ready. I figured that's the time to stop.
All right, good luck, everyone. We'll see you in a minute.
Cool. Where shall I go?
Let's hold on just until we make sure everybody makes it into a room and then we can.
Okay. Oh, it's when everybody has a green dot. Is that when they're there?
Yeah. Yeah.
Ah, I see.
So can you see?
I'm learning something every day.
Can you see what I've named the rooms? Okay, why don't you pop into locations and let them know they've got locations. And then
Oh, they will love me for that.
I'll pop, and maybe you can just hang out there for a little while and I'll pop into severe fragmentation and subpopulation and let them know what they've got. And then what time should we come back? I think we're a little bit behind now, but we had quite a buffer. So
Yeah, because I talked too much. Just after midnight, so after your 10
okay.
or whatever it is where you are. 5 past, maybe something like that.
Okay.
Cool. Bye.
Bye.
I need to answer.
Hey, everyone.
Hello. Oh. Hello.
Hooray, I made it. My laptop is very slow to go from one room to the other, into rooms and out of rooms again.
I got stuck in one of the rooms, which was confusing. I don't know how I got stuck there, but anyway.
Yeah, I seem to be stuck between rooms quite a bit. All right, let's see if everybody else is coming back. We've already got the locations room. Even after, I don't know, like 2 years of being chucked into breakout rooms, I'm still not used to being stuck in these alternate universes where I'm like, what's happening? It's all too much. Right, is everybody back? Can we see this somehow? Yeah, everybody's back. Right, okay, let's start our little report back. Let's start at the top and call on the people who were in the breakout room discussing locations. Would you like to share your example with us that we just talked about?
Okay, so I've been chosen to present my example. So I'm going to present on a species called the Krisangi leaf insect, which is an endemic species to Singapore. It's found in the mangroves and a particular mangrove location in Singapore, which is pretty small. So the population has declined, and one of the main threats is the loss of a suitable habitat because of the loss of its food plant. So that's one of the major threats right now that cause the population to drop to less than 100 numbers. Secondly, will be poaching because of the pet trade. We're starting to see a number of locals having this species at home. And then also, one of the current threats right now is the sea levels rising, and potentially it's been projected for the sea levels to rise even more. So there are 3 existing threats for this species.
And what's your conclusion? How many locations are there for this species based on these 3 threats that you could consider?
3 locations. Would that be right?
It depends again entirely over what area any of those threats operates. So if your sea level rise, if it's really a coastal mangrove, which they mainly are, and sea level rise is already impacting the species and it's over a restricted area, it could be, for example, a single location, it could be 2 locations. It's not the number of threats, it's the area over which a threat impacts the species that you need to consider.
Okay.
If it's a small area and people know where it is, and poaching is quite, and there's like, say, I don't know, a little town nearby or stuff, and it's easy to get to that whole area and just happily wander around and impact all of the species, all of the individuals in that population if you wanted to. It could also be a single location based on poaching, or several locations because of poaching, for example. Any other questions for group 1 locations? Everybody else is stunned into silence. Yeah, so when we define the number of locations, really the most important thing to remember is that it's a geographically distinct area in which a single threat event can rapidly affect all individuals of the species. So if you have it in a restricted area where you have a threat that could rapidly impact all of the individuals of the taxon, then you would have a single location based on that threat. If it's more widely spread, you might have 3 locations because you would need 3 separate threat events occurring throughout of this particular threat, say of poaching, where that would then make up your 3 locations. Does that make sense?
Yeah.
It is by far the most complicated concept of the red list, and it still breaks my brain on regular occasions. So just saying, this is the room with locations. That was really, that's the toughie. Any more questions for group 1? If not, we go to severe fragmentation, another favorite. Who would like to present their example? Ooh.
I will. I've been volunteered. So I was talking about this example of a bee that I was working on, Varrohalea selectivus. So it's the only species in its genus in Australia, and there's one other species in Papua New Guinea. It hadn't been found for 100 years. So as part of a different project, I was trying to relocate it as well. Definitely a side project. And so this map is showing the 3 places where I've found it so far with the green, and the places where I haven't found it on this particular host plant in black. And we were talking a bit about, well, if more than 50% of the population occurs in severely fragmented and small isolated patches, you might be able to use this, but I'm not really entirely convinced. So for example, this one here, Athene, is like a 1 hectare rainforest patch in the middle of like cleared rainforest. This one, Youngla, is actually, this blue is more or less a mountaintop, surrounded by cleared land and like lower land. But Kuranda here is contiguous with like this larger dark blue. It's only been found in tropical or subtropical rainforest so far. So I'm not entirely convinced that this one is severely fragmented, but it's possible. And this is what we're talking about. I'd love to go back and sample more if I lived closer by. Yeah, if people have questions, I'm happy to try and answer them.
Cool. Do we have any questions, any remarks for this group? It's great that you instantly mentioned but I'm not sure if more than 50% occur in really small isolated patches, because that's precisely the thing that this is all about. So this is the thing that you really need to consider. So that's great. Also, happy memories of that area. That's where I saw my first ever platypus. There you go.
Oh, cool. I think it's where a lot of people do.
Yeah, it's apparently a very good spot, Youngla.
Yeah.
You heard it here first. Any other remarks? No, are we all happy? We're all a bit tired. That's what's happening. Group 3, subpopulations.
Yeah, in this paper, I didn't say anything about trying to list it. I said that more work needed to be done. Like I said, I wasn't convinced that I knew enough.
Cool, excellent. Anything else to add, Anna?
No, I guess I'll just say that I struggle with severe fragmentation quite a bit, because one, you need a dispersal distance, which, you know, even if you don't know it for your exact species, you can use a closely related species, maybe something in the same genus. But you also have to prove that the small isolated populations are not viable. And that's a little bit more difficult. And what I usually end up relying on is maybe there's a species that lives in small habitat fragments, and some of those subpopulations have started to sort of disappear. So maybe that would be a way to argue, look, when they're small and fragmented like this, they aren't viable and they're not recolonized. So even if you don't feel like you have all of the data you need, sometimes you can still make a reasonable argument. You just have to justify it thoroughly. But again, yeah, it's a tricky one, especially when we don't always have the data that we need.
Yeah, I find the viability one of it just the most difficult bit of it, because that seems to suggest that I know what makes a viable population of something, and I'm not even sure quite where they occur, and it's very tricky. The dispersal thing, I think it was much easier when we did dung beetle assessments where some of them were flightless, and I was like, well, they're not going to get from A to B, that's fine. But again, it was like in these forest fragments where they still occurred, well, they might still be viable, to be quite honest. So again, it wouldn't quite fit. So it is a really difficult concept. And suffice to say, I prefer locations over severe fragmentations, which tells you everything that you need to know. Okay, room 3, subpopulations. Who have we got volunteering to tell us something about subpopulations?
Okay, I'll do that.
Awesome, thanks, Piers.
Okay, I'll introduce subpopulations. Our understanding of subpopulation, it can be defined as geographically distinctive groups in a population that has been separated by geographic barriers or different landscapes that reduces or limited the demographic and genetic exchange between each group. Definition of population. And here I have 2 examples of subpopulations. Yeah.
There was a lot of noise in the background. Was it at your end, Piers?
No, it's not me. I'm in my study.
Good, it stopped now, so cool.
As you can see here, this is the distribution pattern of a clubtail swallowtail, Lossaria dubodae. Actually, it's several subspecies identified through morphological and genetic characters from northern India, northern Myanmar to across Indochina and some of the islands. At least 4 subspecies in Indochina. They're separated not by mountains or river valleys, but by different landscape characters. You can see this area in Indochina, southwest Thailand and Cambodia. There is no forest, but only swampy savanna in this area. And the Lossaria is a typical forest species that feeds on a certain group of Aristolochia in that area. And its host plant cannot survive without a forest canopy. As a result, different landscape characters separated the species into several groups. This is one example of subpopulations separated by landscape characters. Also, I have another example. This is a projected map of an Apollo butterfly in southwest China. This corner here is northwest Yunnan with several high altitude mountains. Between them are several low altitude river valleys. For instance, here is Irrawaddy and Salween and Mekong and Yangtze River. These butterflies can only live on mountain ridges with their host plants, and they cannot go down into the valley and climb up another mountain. As a result, even it's the same species, it has been separated into many highly fragmented subpopulations across its range, especially in Southeast Tibet. This is an example of subpopulations created by geographic barriers. These are my sharing. Thank you.
Thank you very much, Piers. Any comments?
May I give another example?
Sure.
Okay. Excuse me for the noise. Actually, I am on my way to my workplace, so some noise is there. Actually, I am giving an example from one tropical deciduous forest fire situated in eastern Indian state, one of the eastern Indian states. So there is a forest fire, deciduous forest fire, and they are confined some kinds of Mussolini species. They go through their breeding phase during the pre-monsoon, and their larvae are semi-aquatic in nature, loving the muddy places, muddy patches like this, but not those are exclusively aquatic phase of larval stages. And beside the forest bed, within 7 to 8 kilometers, there is a huge dam, natural water reservoir, and on the bank of that dam, there is grasslands, pasture lands, invaded by a few invasive plant species and affected by grazing activities, etc., and other anthropogenic villagers' life activities. There we found some Mussolini species which are completely different from the forest ones, and their larvae are aquatic, exclusively metapneustic larvae. Again, inside the village, we got few other kinds of Mussolini species. And the village is, distance of village from the forest is within 10 kilometers. So those could be considered subpopulations.
Sorry, it's like at times the sound was quite bad. I don't know if it's my laptop speakers that are kind of introducing a lot of noise to it, or whether that was the connection. So I couldn't quite hear all of the details. I think the most important thing to remember and the most important thing to remember about subpopulations is that there really is essentially very little movement from one subpopulation to the other. There's essentially this barrier. So explaining that there is this barrier and what constitutes this barrier and how the species can't make it across this barrier by whatever means is kind of the real key point to subpopulations and to the definition of subpopulations. Anna, do you have anything else to add?
Not necessarily, but I do think that's important to remember, because if we think about something like the monarch butterfly, we assessed it recently, and they kind of consider that there's two different subpopulations for the migratory subspecies. There's the eastern migratory population and the western migratory population. Well, it turns out there is quite a bit of genetic mixing, because some individuals from Arizona end up in California some years, and some years they end up in Mexico. So in the IUCN definition of the word, those are not two separate subpopulations, even when more generally speaking, they are considered separate subpopulations. So just keep that in mind. It's a very specific definition that they're looking for.
Yeah, correct.
Yes.
Excellent. There's a question from Evelyn.
Hi, my question would be to Piers about the Apollo populations. This map you showed was actually showing a mountain range, and then from one mountain range to the other. You said that the butterfly cannot fly down the valley and then up, so they have to be segregated subpopulations. Is there any scientific study supporting that, or is it more of an observational findings?
Okay, thank you for your question. For Apollo butterflies, the restriction appears to be more stronger in low latitude mountains, such as Northwest Yunnan, than the Tibetan plateau. Because in low latitude areas like Yunnan, the valley area is in all tropical climate, and up in the mountain ranges, they are temperate or subalpine. And Parnassus butterflies are very localized, depend on its host plants. So they are literally reluctant to move far distance for searching for new habitats, especially in Yunnan, where the low altitude areas are very unsuitable for them because it's too hot for them. But in Tibetan plateau, the situation might be slightly different because the base altitude of that plateau is already favorable for many Parnassus butterflies. So they can migrate from one mountain ridge to another by, for instance, 3,000-meter valleys in between. It's not a problem for a few species, such as Parnassus imperator and Parnassus orleans. However, for species like restrict high altitude ones, like Parnassus labiarii or Hidai, they cannot move as well. They are restricted on that mountain ridge. I hope that this helps.
No, that's very informative because we have the species Parnassus apollo in Turkey, and among the mountain ranges, I would assume that they would move and make a longer subpopulation. And for me to identify different populations of the same species, there should be a huge distance. But now that you explained that the valley bottoms are really different tropical environment, it makes more clear for me that they actually don't travel that far. Thank you. That was a good example.
Thank you.
Yeah, and the questions that you ask are precisely the kind of questions that a red list reviewers would ask. It's like, oh, but why is this? Why is this? What is it based on? Or also what we would discuss in red list assessment workshops. So if we have actual workshops where experts come together, these are the kind of questions that we ask simply to justify whether we've used these terms correctly. So that's really, really good. There was a question from James. Yeah, I mean, if you do have genetic data to support that there's isolation, then you've really hit the jackpot. Because then I'm like, yes, you can kind of show that ultimately these subpopulations are isolated, and I would love that if we had that for all of the species that we need to assess. That would be beautiful. Cool, excellent. Right, that wraps up this part of the show. Shall we go straight into? I know we had a little bit of a break planned, but we've overran because we had too much fun.
Yeah, how are people feeling? We are running behind, but I can probably get through the last few presentations in about 25 minutes. So maybe we just take a 5-minute break. Is that enough for everybody? All right.
Yeah, sure. Good.
So we'll see you back at 35 minutes past the hour.
Yeah, cool. I'm so tired.
No, I'm tired, but it's so much later for you. I'm very impressed. You're doing well.
I started with a 7 a.m. meeting this morning as well, so I've been going for a while. So you know.
Wow, yeah. I hope you got your nap in.
I did.
Good. So yeah, I think we are gonna run out of time for the exercise today. So I think I'll just send people home with it, and we can review.
Yeah, we can just send it and we can review it tomorrow to start off with or something.
Yeah. So I'll try and get through the last 2 presentations, but they should be pretty quick. So.
Excellent.
All right.
It's always the terms used that always takes the longest because there are questions.
Yeah. Yeah, it's good to it's good that there are questions. It helps clarify things right from the get-go.
Yeah.
All right, I'll be right
Precisely.
back. I'm gonna take a short break.
You're allowed to.
On my way.
Okay, I'm going to go ahead and get started. You may notice I just dropped a few links in the chat. The first one is a Google form, basically just kind of a feedback form for you to ask us any questions, anything that's still not clear. And then tomorrow, first thing, we'll address any questions that you have. We also just want to make sure everything is going well. So please give us feedback. The second one is the exercise for one of the presentations I'm about to run through. And we're not going to have time to go over the answers today, so we'll just kind of send you away with it. But we can go over it first thing tomorrow so that all of the concepts are crystal clear. So the first little short presentation I'm going to run through is just on the IUCN Red List categories. So there are 9 IUCN Red List categories. So all of the world's species can be placed into one of these categories. So the majority of species, especially the majority of insect species, currently sit in this category, not evaluated. So these are just species that are not yet listed on the IUCN Red List website. So these are all species that haven't been evaluated. There are also species that have been evaluated, but we really don't have enough data to determine whether or not they might be threatened. So all of these species end up falling out as data deficient. So it's really important to keep in mind, especially for insects, that both of these categories, not evaluated and data deficient, are really important because they sort of highlight areas where we need to maybe focus some of our assessment attention. I know a lot of people with invertebrates will say, well, I can't even assess that species because I don't have any data on it. And that might be true, but it's still very important to get that species assessed, even just to highlight the fact that it's data deficient. Because then if somebody comes along and they want to do some sort of monitoring project, they know exactly where to go and what species to look for. So then the remaining 7 categories are used for species that do have sufficient information available. So with relatively low extinction risk, we've got least concern. These are the species that are currently at fairly low risk of becoming extinct. So these are usually widespread, usually abundant, but it's important to keep in mind that this might also be used for species that maybe have a restricted range, but there are no current threats. So a species like that, you might intuitively think it's threatened just because it has a small range, but if you can't point to a single threat, it'll come out as least concern. This also applies to widespread species that are undergoing very slow population declines. So in these cases, the current extinction risk is relatively low, even if some conservation measures might be warranted to stop it from slipping into a higher threat category. But just because it's least concern doesn't mean that it's not in decline, it's just not hitting the certain threshold for decline. So then we've got near threatened species, and these are species that have a slightly higher risk of extinction, but are not yet threatened. And these species usually meet some of the thresholds under the criteria that we'll go over tomorrow, but maybe not all of those thresholds. So this is just a way of highlighting that this species is currently not in a threatened category, but it wouldn't take much for it to get bumped up to a threatened category. So moving higher up, we've got our 3 threatened categories: vulnerable, endangered, and critically endangered. And these highlight those species at the highest risk of becoming extinct. And collectively, these are our threatened categories. And it's important to note that critically endangered also has this possibly extinct flag for those species that maybe haven't been recorded in a long time despite survey efforts. So keep that in mind. Possibly extinct is not a category, but there is a flag for it so that we can identify those species when we come across them. Next, we've got extinct in the wild, and that just highlights species that are now only known to exist in captivity, so zoos, aquariums, and botanic gardens. This also might be populations or species that are introduced to areas outside of their natural range that are no longer found in their natural range. They'll be considered extinct in the wild as well. And then, of course, the worst case scenario, we've got our species that are currently thought to be extinct. So this is when all of the captive individuals have died, and we no longer think the species is present in the wild. So as a species moves through these threat categories from least concern, its extinction risk increases until it finally reaches extinct. So the IUCN tries to ensure that species are reassessed at least every 5 to 10 years, depending on the resources. And through this reassessment process, species can change red list category. So there are a couple different reasons why a species might change from category to category, and not all category changes reflect a genuine change in status. So it's really important when you're reassessing a species to think about why it might have changed. So we've got non-genuine changes, and this includes things like new information has become available since the previous assessment. This is probably the most common kind of non-genuine change. Maybe there have been some taxonomic changes, such as a couple species being lumped together or others being split apart. This is a non-genuine change. Incorrect data used previously. Unfortunately, sometimes the previous assessment maybe had access to data that wasn't quite right. Sometimes the categories and criteria are revised. I think the last revision was in 2001. So if your species was assessed prior to 2001 and it's now being reassessed, you might want to double check whether the criteria has changed since then. Sometimes the knowledge of the criteria can cause a non-genuine change, which is why it's really important for everybody who's writing red list assessments to fully understand the categories and criteria so that we can prevent this sort of non-genuine change from happening in the future. And then there are genuine changes in status. So these are species, it's really important to highlight if it's a shift to a higher risk category, you might want to highlight that to prioritize conservation action. And best case scenario, sometimes we see a genuine change where our species is actually doing a little bit better and maybe shifts to a lower threat category.
Everyone has a question.
Oh, yeah.
Sorry.
I'm sorry for interrupting. One of the things that we had while writing our assessment was that we had a species defined. And then later with the taxonomic statement, they said that this is not a species anymore. This is a subspecies. So what do we do with that assessment? Do we just delete it or ignore it or remove from the system? What happens to that situation? I'm sure you're having this problem a lot.
Yeah, I mean, the assessment will be in the system forever. It'll be accessible forever. And in that situation, what you could do is you could write a subspecies assessment, because essentially you've already got all the information you need. You just take what was written for the species and assess it as a subspecies.
But isn't it required to first assess the species, then the subspecies?
Yeah. Yes, it is. So yeah, you might end up having a little bit more work to do, but that would certainly be an option.
Okay, thank you. Sorry for interruption.
Oh, no worries. So for genuine changes, there are certain rules outlined in detail in the Red List guidelines. So where a species is genuinely deteriorating in status, it must be uplisted to a higher risk threat category immediately. So even if you don't come in and say, oh, well, this species hasn't hit its 5 or 10 year reassessment cycle, so we'll just wait, you assess it as soon as possible. But for species that are showing a genuine improvement in status, we want to be sure that this improvement is going to continue before we downlist a species to a less threatened category. So they've put a 5-year time lag on just kind of as a precautionary measure. So in this example on the screen, a species is initially assessed as critically endangered, and then 2 years later, it shows significant improvement that might downlist it to endangered. But the species can be reassessed, and the supporting information attached to the assessment can be uploaded to support this change. But the category itself should remain as critically endangered for at least 5 years, just so that we can make sure that that trend continues. Because either the status of the species will continue to improve, so then once we hit that 5 years, we can reassess it. But sometimes, in some cases, the improvement might just be temporary and the population might begin to deteriorate again, or maybe it was just a little bit of a fluctuation in population size. So just as a precaution, we want to make sure that it definitely has shown genuine improvement. So moving now on to data quality and uncertainty. I apologize if I go through this really fast. These are pretty important concepts to understand, but if they're still a little fuzzy after today, we'll definitely go through them all tomorrow as well. So dealing with a lack of high quality data. This can be pretty daunting to assessors at first. Although quantitative thresholds are used to determine which threatened category a species can be listed under, the system definitely has room for quite a bit of uncertainty in the available data. So there's a lot of situations where we really have no direct data at all, but we're still able to make inferences about what the situation might be. So it's clear that if we were to rely on lots of detailed research data that is 100% certain, we would never be able to assess very many species at all. So there's ways of dealing with a lack of data and uncertain data in red list assessments. So if you look at your handy summary sheet, which hopefully you'll all kind of make it somewhere accessible over the next few days, you'll notice all of these terms that pop up over and over again: observed, estimated, projected, inferred, and suspected. And these terms basically just refer to the quality of the information that's used in the assessment as defined in the red list categories and criteria. So the full definitions for each of these terms appears in the guidelines document that you'll want to become familiar with. But we'll go over them now quickly. So observed data is the highest quality information we have. It's based directly on well-documented observations of all known individuals in the population, for example. So if the entire global population of a species occurs in one small area, and all mature individuals within the population are counted each year, then the assessor would know for certain that the rate of decline, they would know for certain what the rate of decline for that species is. And you can imagine for something like a large tree, it's quite easy to count, especially if there aren't very many individuals over a small area. It's easy to count all of the mature individuals from year to year. But for something like a butterfly or any other insect, this is a challenge, and we will probably never have an observed population size or population decline. So estimated information is generally based on some sort of scientific study. So for example, if regular surveys are carried out at a specific site within the range area of a species, the data gathered from these surveys can be used to estimate a rate of decline across the whole population. So in this example shown, the method results in an estimated reduction of 65% between 2005 and 2008. And estimated information is based on calculations that involve assumptions. So that could be statistical assumptions about sampling methods used, biological assumptions about the relationship between an observed value, say an index of abundance, and the variable of interest, which in this case would be number of mature individuals. So all of these assumptions that you will be making to estimate some of these parameters should be really well justified in the documentation that goes along with the assessment. So projected information is basically the same as estimated, but the variable of interest is extrapolated in time towards the future. So in the example shown, it's the same as the one on the previous slide. If additional information is also available on the causes of decline, so if maybe we have habitat loss and the likelihood of the threats continuing into the future hasn't changed, then a population decline model can be created and extended into the future. So in that model, past decline is estimated from the collected data, and future decline is projected based on the threat continuing as it has in the past. So for projected information, the assessment should always include a discussion of the method used. So this is, basically just a justification of statistical assumptions or the population model used, and a justification for extrapolating current or potential threats into the future, including the rates of change. Inferred information is one of the weaker data quality types, and it's based on indirect evidence or variables that are indirectly related to the variable of interest, but are in the same general type of units. And this same general type of units is important to remember because it'll help distinguish between this kind of data quality inferred and the next one that we'll talk about in just a minute. So keep that in mind. So for example, we might not have any information at all on past and current population sizes, but if the species is utilized, there may be trade data that suggests the population has declined in recent years. So in this example, we're looking for information about the population trend. And although we don't have any direct data on population size to give a certain answer about exactly what's happening to the population, maybe we have access to trade information that does relate to numbers of individuals of the species. So while we don't have enough data in this case to estimate a population decline, we can infer that a continuing decline in the number of individuals based on the change in trade figures is happening. So we're assuming that the decline in the presence of the species in the market reflects an actual population decline rather than a change in something like harvesting effort or target species. Maybe the market forces are changing. So that's why this isn't quite as high data quality, but it usually can at least tell us something about the situation. So for suspected This is the weakest type of data quality, and it's based on circumstantial evidence. So this could be surveys of local people living within the species range who maybe say the species is utilized less or more, or maybe they don't see it as much, this kind of thing. Or it could be based on variables in units different to the kind of variable we're measuring. So an example of this is a percent population reduction based on something like habitat loss. So in this example, the suspected rate of population reduction is based on the rate of habitat loss. So note that evidence of qualitative habitat loss can be used to infer that there is continuing decline in habitat quality, or AOO, but you need evidence of the amount of habitat loss to use for a suspected population reduction at a particular rate. So if you don't have that amount of habitat loss, so maybe this might apply even if you don't know how much of a habitat specialist your species actually is, then you might not be able to suspect a population reduction at a particular rate. So again, there are quite a few more assumptions involved in suspected information, and these should be really clearly outlined in the assessment documentation. But it does mean that even if you don't know everything definitively, you might still be able to suspect or infer and get yourself to a little bit more information. So dealing with data uncertainty, uncertainty can come from a few different sources. It could be that there's natural variability as a species' life history and environment change over time and space. It could be because the vagueness in the terms of the definitions used in the criteria. And it could be from something like measurement area. And when there's uncertainty with the data itself, a plausible range of values may be obtained. And this happens quite a lot. So in these cases, what should the assessor do? We're going to record the range of possible values in the assessment documentation based on the available data. We're going to state the range of potential categories the species may qualify for. So for example, if our population size is between 100 and 410, and that comes from a couple different estimates, and we're really not sure which one is more reliable, then we can state that range, and we can say that at this range, our species might be considered endangered or vulnerable. And then we're going to rely on some of the other information that we have on things like population and habitat type, and maybe what threats might be ongoing to select the most plausible category from that range. So for example, if we think there's a lot of threat pressure, we're going to be more cautious and lean towards the endanger side. However, if we're like, oh, that one threat doesn't seem all that plausible in the next few years, maybe we'll lean a little bit more towards vulnerable. So there is some discretion in there, although the attitude that the IUCN recommends taking is a low dispute tolerance. So that means when there's some uncertainty, it's better to err on the side of caution, and maybe in this case, we go with an endangered listing instead of vulnerable. It is important to note, though, that when the range of values results in a really wide range of potential categories, so maybe it could be least concern or it could be critically endangered, in these cases, we're going to end up falling out as data deficient, because in we just really don't know enough about that species to make any assumptions either way. So that's pretty good timing because we're just hitting the top of the hour now. And this is the exercise I mentioned, I left in the chat. So if you've got some time before tomorrow, please look over it. It's only 5 multiple choice questions, I think, and hopefully it'll get you to think about these concepts a little more, and we'll go over it. And please also fill out the form in the chat. Tomorrow, like Moni mentioned earlier, we'll be digging into the criteria one by one, and we'll be pausing between each presentation to get into some breakout rooms again and do some exercises. So we'll start looking at some actual assessments, and it should be a busy day, but it'll be fun. Any other questions?
You'll hear our voices less often tomorrow, hopefully.
Yeah.
Yeah, I'm wondering if I need to miss one of the days, is it okay to just watch the recording?
Yeah, it might not be quite as helpful because you won't get to participate, but it's certainly better than nothing. Especially if you do it, if you're able to watch the recording before the next session, because then you're at least caught up, because you can get left behind quite quickly if you miss too many sessions.
That's what I was thinking I'd do, just have a full day.
Thank you. Bye.
Yeah, thank you, everybody. It's great to see you all, and thanks for coming out. We'll see you tomorrow.
See you tomorrow. Thank you. Thank you for your.
Thank you.
See you tomorrow, everybody.
Yep, see you tomorrow.
Thank you very much.
Thank you. See you tomorrow.